From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 34, recorded on August 14th, 2018. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Greetings from the smoke-tinged Elde Lab studios.
Yeah, get some forest fires out there? Is that what's
Yeah,
happening?
that's right. Exactly right. It's been a somewhat brutal wildfire season so far, and so I'm looking out from my office and pretty hazy across the valley. We've had some local fires, but even some of those massive fires out in California the smoke has migrated here to the western Rocky Mountains.
Yeah, you do border on California, don't you?
Well, with Nevada in between.
Oh, forgot about that. That's right, the Nevada, the angular state there. Yeah.
The West Desert, yeah, that's right.
It nestles right into the crook of California there. And then we got Utah. So the smoke can make it all that way, huh?
Oh, yeah, no problem. And I heard a rumor that it had made it all the way to New York, but have you seen anything?
I haven't. No.
No?
I haven't seen or smelled any smoke here. Not from California anyway.
Fair enough, yeah.
Yeah. I heard somewhere it's been burning for 2 years now in California.
Yeah, and that's right, on and off. And I think it's traditionally just the very start of wildfire season for this year, but it's off to a terrible start where I think the biggest forest fire, wildfire recorded is now continuing to burn, the Mendocino complex fire. So that and a handful of others are really changing the landscape out there and influencing ours right now.
Yeah, it's too bad, but that happens. That's what happens.
Yeah, it happens. It hasn't been super bad here so far. And in fact, I wanted to mention to have you tip off Dixon Dupalmier to pass along to him. I was just out fly fishing with some success on the West Fork of the Duchesne River, catching Colorado cutthroat trout. It was really fun.
Wow. West Fork of the Duchesne.
Yeah.
I think that's where he is today. Because he's not in, he's probably fishing.
He's out fishing, gone fishing. Yeah, good for him. I'll send along a little photo here that maybe you could pass along.
Yeah, be happy to do that. We've had a lot of rain here, and he says that you can't fish when there's so much rain. Yeah, because rivers flood or whatever. I don't know what happens.
Yeah, they can flood, and what can happen is the flies get sort of blown off the trail, and so it's hard to run a fly fishing sort of lure.
Nice picture, very nice.
Yeah, that was a fun one.
It's a little. Is that a brown trout?
So that's a Colorado cutthroat.
Cutthroat
trout. Yeah, yep. Really interesting fish. Beautiful fish. So if you turned the fish kind of underneath, and we're looking at the bottom side of the gills, there's a red stripe that gives the trout its name, the cutthroat. Yep.
I have to show it to him.
Yeah, please do.
He caught a bunch of different kinds in Montana last summer. I was with him on a boat float, and I think maybe cutthroats were some of them. But they have cutthroats up there in Montana?
Yeah, they do. And there's several different varieties. So in Utah, I think there's 3 varieties, the Colorado, the Bonneville, and then also the Yellowstone, which is what I think you would find in those waters where you guys were last summer. Mm-hmm.
Now you have 2 kinds of sunglasses here. I noticed.
Guilty as charged. Yeah,
and what does the shirt say? I can't read it.
Oh, yeah. So that's a shirt for the old science mill. We've talked about this tiny bit on some previous TWiEVOs. This is a interactive science museum down in the hill country of Texas that I've been involved in the last few years. And so the sunglasses, maybe we could put this on our website. This is, sort of a fashion statement, as well as the polarized sunglasses that you can really see into the water. and actually see the trout swimming around as you sort of
cool.
decide where to throw your flies. Yeah.
Otherwise, it's all reflective, right?
Exactly right. So having those polarized lenses can really make a difference.
Well, you don't have any waders, man. You just have boots, little big shoes.
Well, it's the hot summer season here, nearing triple digits
low water,
on the Fahrenheit scale. With pretty low water. It's been a dry year, so kind of adapting to be as comfortable as possible out there.
Yeah. All right, cool. Looks great. I'll pass it on to Dixon.
Yeah,
I'll ask him if he knows what kind of fish it is. I'm sure he does.
Give him a quiz. Yep. He might have mentioned the West Fork of the Duchesne to me last time I was in New York at TWiEVO Studios there. Yeah.
So what do we have for our listeners today, Nels?
Yeah, so I have a paper that I'm pretty excited to share. This just came out in the last week or so, and I think I did see some press coverage of it. I think it's an interesting collision of evolutionary biology, evolutionary genetics, with some environmental policy and even politics, as we'll see as we dig in deeper. But to begin, the title of the paper is Ancient Convergent Losses of Paraoxonase 1 Yield Potential Risks for Modern Marine Mammals. This paper showed up in Science magazine a week or so ago. First author is a postdoc, Wynn Meyer, and she is in Nathan Clark's lab at the University of Pittsburgh, one of my favorite labs out there, I'll say. I know Nathan from back in the day. So when I was doing my postdoc at the Fred Hutch in Seattle, he was a graduate student in the Genome Sciences department at the University of Washington. And then Nathan went on to do a postdoc at Cornell, if I'm remembering correctly, and then probably for the last 6 years or so, has been running his lab at the University of Pittsburgh. And, just to say quickly, I think Pittsburgh is really emerging as a powerhouse of science. I'm just thinking about both in virology, so some of our colleagues like Terry Dermody, Carolyn Coyne, and also notable for a growing crew of evolutionary biologists and geneticists as well. So they just launched, in the last year, a new center for evolutionary biology and medicine. There's a fellow called Vaughn Cooper, who I just actually ran into in Park City, nearby my local location here. There was an evolutionary medicine meeting about 3 weeks back, and Vaughn was here for that. He's also helping spearhead the Center for Evolutionary Biology and Medicine. Sort of an interesting collision between, evolutionary biology, which traditionally we don't think of as applied to medicine, but a growing population of folks who are sort of interested in bringing the two fields together in new ways as the impact of the things we're learning from evolutionary approaches, especially in this post-genome era, could have real implications for thinking about personalized medicine, health, and extending, as we'll see today, into sort of the ecosystems around us.
I met Vaughn when I visited Pitt a couple of years ago. Mm-hmm. He's a sharp-tongued man. He can be sharp-tongued.
Yeah, Vaughn's an interesting guy. So he did his very diplomatic description there. So he did some of his training at Michigan State in Rich Lenski's lab, who's noted for these long-term experimental evolution courses with E. coli, stretching back, right? This great sort of seemingly simple experiment where you take 100 microliters of E. coli, split it out into 12 flasks every day, and just kind of keep doing that. And tons of insights into how these replicate strains have adapted. Vaughn came out of that crew, contributing to some of that work and then extending it in new directions as he's going forward, including with some bacteria that have clinical relevance. And how do these things adapt? And how might they adapt sort of under different conditions? And can we apply that knowledge to ways to combat things like antibiotic resistance and related issues in medicine?
Mm-hmm. Mm-hmm. He's an interesting fellow. Mm-hmm. I don't mean to say he's not interesting, but he definitely speaks his mind in a sharp manner. I remember I was in the airport once here at Newark. Pitt buys ads on those TVs that are hanging around the airport, right?
Oh, sure, yeah.
And they go through, huge NIH funded site, blah blah blah, and then every now and then a few people I know pop up on the board, on the TV, as part of the ad. So I tweeted once, why do I have to see these ads for Pitt while I'm at the airport? And I said, why don't they give the money to the labs for research instead of spending it on this? And he chimed in. He was on Twitter at the time.
I see.
And he said, well, maybe it's a good investment. I said, well, how would you know? So we got into a little argument, but I think I still see them there to this day. I go in there and I see Graham Hatfull up there,
Oh, yeah. Yeah, Graham's great. Yeah.
And a bunch of other people.
Mm-hmm.
And, it's just got some thunder outside here, man. Gonna have a thunderstorm rolling through.
Cool.
So I don't know. I just think they should give it to research, and I'm sure they could use it. And what the hell is advertising their researchers on the airport monitor in Newark Airport going to do? Hey, we had a nice chat, but
yeah, good question. Maybe we could try to convince these folks to redirect some of these resources. They could sponsor TWiEVO, for example. We could read a quick ad.
That would be self-serving. This one, give it to these guys who are doing research. But it's funny, he didn't want it. He said no, it probably could. I had dinner with him when I was at Pitt visiting. I gave the Julie Youngner lecture.
Oh, right.
In January of 2016. And I had a nice dinner with him on Friday night.
Yeah.
He's a good guy.
Great.
Just very, he's opinionated for sure.
Sure. Good energy out there. Graham Hatfull is another example. Yeah, growing contingent crew. Okay, so getting back to kind of the paper at hand. So the Clark Lab has been, over the years, actually building several computational pipelines to track both coevolution and then convergent evolution. So this idea of convergent losses is already even in the title of this paper. We'll dig in on that in a moment. Just wanted to mention on a personal note, we've had some fun teaming up with the Clark Lab on thinking about coevolution of some of the, in the evolutionary history of some of the immune nucleic acid sensors, these things called CGAS, OAS1. These are proteins that can sense the presence of either double-strand RNA or DNA in the cytoplasm of the cell. It's sort of like blood in the water for the innate immune system, and to trigger an immune response. And in some of our work, we teamed up with Nathan to use some of his computational pipelines to look at the idea that OAS1 and CGAS might be evolving sort of in similar ways, in both among sort of a sampling of different primate species, including humans. And then one of the things that was really fun that we uncovered was, if you look at the sequences of OAS1 and CGAS, they're completely different. But if you look at the molecular structures that have been solved for the two proteins and you put them overlap them or overlay them on each other, they're almost exactly identical.
Cool.
And more than that, in one or two of the helices, there are 4 or 5 amino acid residues that have independently been evolving so rapidly with all these fixed substitutions, both in OAS and in CGAS, sort of an potential example of convergent evolution.
So what does it look like, the protein? Is it a single lobe or multi?
Yeah, so it's good question. So it looks like something like 2 lobes. So there's an enzymatic active site. And what happens is double-strand RNA or DNA, depending on the protein, is bound in sort of one of the lobes. This causes a small conformational change so that the enzyme becomes active. And what both of these proteins do, it's a very different covalent bond that it forms, but they both take small nucleotides, ATP or AMP, and form chain linkages. So for CGAS, they form these things called cyclic GMP. And for OAS1, it forms an oligo, a 2' 5' oligo. And both of these molecules are different, but they both are similar in the sense that they act as secondary messengers to then trigger the action of other proteins. For CGAS, you end up triggering an immune response through the STING pathway, which upregulates the expression of hundreds of these so-called interferon signaling genes, or ISGs. And for OAS1, you activate an RNase that is otherwise sort of quiet in the cytoplasm called RNase L. It binds these modified nucleic acids and starts chopping up RNA wholesale, and that's thought to be an immune response. You're willing to sacrifice an infected cell to try to save the organism. So the idea is you have similar features that have sort of independently evolved.
Yep.
And that's this idea of convergent evolution, sort of one of the, it's come up in TWiEVO a little bit, but certainly one of the main concepts of evolution is this idea that you can have sort of the independent emergence of similar features in species of different lineages. And so one of the most sort of familiar examples of this would be when you see fins in whales and fish. Whales, as we'll actually be talking about for a different reason, are marine mammals that developed fins independently of fish that have always been sort of swimming around in oceans and bodies of water. So the functions are the same, but the evolutionary origins are different. And this is an example of convergent evolution.
Yep.
So speaking of finny critters, or marine mammals in this case, what the Clark Lab at Pitt has been doing for the last few years is taking advantage of this great feature of the evolution of marine mammals, which is, so, if we think back to our kind of early biology classes and this idea that our ancestors actually emerged from the oceans to become terrestrial critters, and then a subset of our ancestors returned to the ocean.
They didn't like it.
They didn't like it. Yeah, that's right. The oxygen was a little too much. And these are the marine mammals. And this has happened independently, or as another example of convergent evolution, 3 times. And so you can see this in phylogenetic trees of if you just sample mammals. And so in this case, the Clark Lab, and this is in figure 1 of the paper, shows a sampling, I think, about roughly 100 mammals. Mm-hmm. And then they're highlighting 3 clades or lineages, clusters of species that are now marine again. These are the marine mammals. And so let's see, what are the 3 categories here? The cetaceans, which are the whales and dolphins, the pinnipeds, which are represent the seals and walruses and some of the related species, and then the sirenians, which include the charismatic manatees and dugongs of Australia. Yeah.
Now, separately, I guess penguins are birds, right?
Correct.
It's about as close as you get to a marine bird, I guess, right? I mean, they do spend a lot of time in the water, but they can also go on land.
That's right. Yeah. And so if you were to include the birds, including penguins, that would be sort of, you'd draw a branch at the root of the tree, the very far left of the tree, to add those in. So what they're considering in this work is actually not as much divergence. But that'd be an interesting question, is whether so we'll be talking about the loss of this this enzyme and whether that extends even beyond the mammals. That's not considered in this work.
But the penguins, I guess, are an example of a bird that went from the air to, or I don't know where you say they live, air, land to air to land, sea, right?
That's correct. Yeah. So terrestrial as well.
Yeah.
Although, as you're saying, airborne. But then, with penguins, again, returning to the sea. So they're actually critters that sort of do both, right? So unlike whales that have really committed
they have.
to the ocean, penguins are kind of a hybrid lifestyle. But you're right in the sense that they've, they no longer fly, and the wings have really adapted to swimming and to do that efficiently.
I'm sure if you did some studies like we're going to talk about today with the birds and the penguins, you'd find some interesting things as well.
I agree. Assuming that, and I'm assuming that so the enzyme paraoxonase, which we'll be talking about in more detail, so the question is, when did that emerge? And was that in a common ancestor that reaches all the way back to the divergence from our lineage from the birds? Yeah. I think that probably does. There is getting back to the idea of these independent sort of returns to the ocean. What's really nice is that if you sort of look at the phylogenetic arrangement, so who's most closely related to who, in terms of the species, you can see 3 very clear events on the tree. So the manatees are closest to the elephants.
Yeah.
The whales are closest to the hippos and cows. And the seals and walruses are pretty close to things like ferrets and sea otters and even polar bears. And
So what's a sea otter? I guess it's a marine mammal, right? But it's also terrestrial.
yeah, right. And so actually in that, the pinnipeds, as we'll see, this is sort of this is maybe a little bit analogous to the penguins in that you have these species that have sort of these hybrid lifestyles, right? So,
Yeah.
Whereas the manatees and the whales and dolphins sort of are full timers in the ocean, seals and otters and walruses spend some of their time or even a significant amount of time on land and kind of have these hybrid lifestyles. That'll actually emerge as a point in the analysis that they do.
Yeah, it turns out that the otters have the lesion that's in this that we'll be talking about.
Yeah, exactly.
It's cool.
Yeah, I agree. So what the Clerk Lab has done really well is, in kind of the big idea of this paper, is to correlate these independent returns to the ocean with actually shared molecular features that might have also emerged independently by convergent evolution.
Mm-hmm.
And so in this case, they're kind of considering a subset of possible changes, genetic changes that have occurred, and that is the loss of genes. We often think about, the birth of genes or the duplication of genes. Here's the reverse. Sometimes less is more. Or if you have something that you're not using, you'll use it or lose it, sort of a scenario from an evolutionary standpoint. And so what the Clerk Lab has done is built some really nice pipelines to go through, kind of troll through genomic data, in this case, predicted gene open reading frames, so to speak, gene coding regions of the genome, and then ask the algorithm or the computer to return cases where it predicts the loss of a gene, or the so-called the appearance of a pseudogene, or the loss of a functional gene. Mm-hmm. And so these can be the appearance of a pseudogene can be diagnosed, or there are different flavors of these things. Some of them are actually creative or duplicative. These are the so-called process genes, the action of retrotransposons that can take the transcript of a existing gene, copy and paste it, so you have two copies. But a lot of these things are sort of dead on arrival and are considered pseudogenes because they don't actually, even though they're copies of genes, they don't go on to encode any functions. Another flavor of pseudogene is just a simple debilitating mutation. So this could be a mutation that causes a stop codon to appear. So instead of reading through the whole transcript to translate a long protein or peptide of amino acids, you end up with something that maybe codes for something really tiny, but it's not functional and it gets lost. It'll cause frame shift mutations that will sort of throw off the whole message so that it's no longer functional. Mm-hmm. And so that's what they've done here, is they've figured out a computational pipeline to sort of pull out cases of pseudogenes showing up, in this case, the frame shifts or the stop codons. And then what they're doing, which is sort of a twist on this, is not just saying, is this happening in one species or even a set of related species, but is this happening independently? And so this might be diagnosed a few different ways that they illustrate in the paper. So one is, again, returning to that phylogenetic tree. The fact that the marine mammals have independently returned to the sea 3 times, when they just do these comparisons between the different groups, the independent groups, they can ask whether the same genes are getting independently insulted with mutations and lost. Mm-hmm. And they don't go into too much, but I think there's a couple ways to diagnose this. One is that you see these mutations showing up sort of in a spotty distribution across the tree, or in this case, independently in those 3 clades. The second alternative, which is a little bit harder, I would say, to diagnose, is in a group of more related species. So if we look at the bats, for example, like they've sampled maybe 8 or 10 different species of bats. The difference between them and the marine mammals is that the bats are all in one clade. They're all clustered together. And so if you just look at a gene being lost, you might at first say, well, I can't determine whether this was in the ancestor of all bats because they're all clustered together, right, or if this happened independently in the species. However, there are some tricks you can do, and that is if you take a really sort of close view on the events, you can start to ask, do the mutations show up independently? So do you have a stop codon in one species at this location and a stop codon in a different location, or different frame shifts? And that might be another way of figuring out why, if convergent evolution has happened, that is sort of the independent acquisition of losses. So you might ask, why do we care, actually? Why are we so concerned about identifying these things independently? And what I would say, whether it's for this paper, as we'll sort of get into the specifics of one of the genes that they lift up for being repeatedly lost, is that this becomes a really sort of useful way of pulling out from all of the genetic changes. And there's just, within the genome era, the diversity out there is literally mind-boggling. How do we know which changes have been important or consequential relative to all of the changes that are just happening as a regular course of mutation that's happening all the time and may not be sort of consequential to the history of our species or the history of life on the planet? And that is where convergent evolution, the independent, sort of almost the idea of lightning striking twice or 3 times or more, now that allows you to sort of focus your attention on these cases where something might have happened. It kind of, helps you prioritize the changes in the genome that might be, because they're happening more than once, something that's really rare. That is a really nice beacon for exciting biology to be sort of hiding beneath the surface.
And that's where the pipeline is important to do all this, right? The computational
that's right.
Pipeline, right?
That's right. And what it does, I think, and they have a nice table near the beginning of the paper that shows the output of what they found. So the pipeline goes in, tries to predict where mutations have led to gene losses independently in the same genes, but in the different lineages. And what they return, sort of their top 10 list, is a set of olfactory receptors, taste receptors, and that's kind of a nice confirmation that the pipeline is working. So you can imagine, and other folks have sort of published related studies, that when marine mammals return to the sea, if you think about that sort of aqueous environment, how you hunt or kind of sense the environment or taste or smell is different in water than it's on land. And so it's not, that's sort of one of these the idea is sort of this use it or lose it, where there have been all these losses as some of these species have adjusted or adapted to hunting and sort of sensing the environment in water, things like echolocation and other specific adaptations that have probably kind of taken over, where the olfactory receptors might be more vital for something like a carnivore that's trying to find prey, or a fox figuring out that there's a mouse buried a foot beneath the snow.
Before, this idea that you don't need to smell if you're living in the ocean.
Yeah, that's right. I think there is still some of that sense. I don't know the details, but yeah, it's certainly not even close to what, and with other sort of adaptive mechanisms coming into play.
Yeah, I guess when you come out to get some air, a lot of these mammals do that, right?
Absolutely. Yeah.
They all do, right?
Yeah.
And they could get a whiff of the weather,
That's right.
But, whales spend hours submerged, right? So
yeah, exactly.
I'm not sure. As you said, there are molecules floating around. Maybe they can still sense them, but it's different from the kind of sensing in air, I guess. That's the difference. Maybe you'd need different sensors than you would in water.
Yeah, exactly right. Yep.
But this is not a mammal, but a shark can sense blood, right? I presume that's some kind of chemosensing.
That's absolutely right. Yep. Yeah, and then a lot of adaptations depending on the niche that they're filling. And so, like you're saying, the sort of carnivores of the sea, like the sharks going after, other fish and prey, whereas you have the vegetarians in there as well that are kind of grazing, and like the manatees fall into that category, for example.
And maybe the receptors are different, right? That's the bottom line from the ones that terrestrial mammals would be using.
Yeah, and in fact, not just the kind of marine environments, but even terrestrial environments. So a lot of different species, humans have given up a lot of probably our ancestral ability to do better detection, whereas sort of carnivores like cats and dogs have retained some of that ability.
Yeah, sure.
And that reflects, the differences in how our lineages have diverged and our strategies for getting resources, for getting food in hunting, or domesticating crops have really changed. And so that's, again, sort of the idea of use it or lose it. There's even a number of some studies and things like fruit flies, where the olfactory receptors are noted for having a really volatile evolutionary history. Seems like there's a lot of flexibility there, that you can kind of gain or lose these things, and it can be hard to pin any function down on just a single receptor. There's a lot of flexibility somehow encoded into the receptors that are picking up on the cues around us and the sense of smell. So anyway, if you look at their top 10 list, their top hit is actually not and I should say that they do a ranking based on a likelihood ratio test. So they're basically setting up the computational pipeline to make a statistical prediction about whether a gene has been independently lost, relative to the possibility that you're just seeing a single loss or a divergent loss in the data set. Mm-hmm. And so it's kind of shooting to the top of the list is actually not a receptor at all. It's an enzyme. The one that's in the title of the paper, which is paraoxonase 1. And so what is this gene that has basically, they have the best statistical support for observing it being lost independently in these three marine mammal lineages. So this is an enzyme in the blood, in our bloodstream, that processes lipids. LDL, HDL, the low density and high density lipids that we think about when we're talking about cholesterol, and other lipids, phospholipids that get metabolized as part of our diets. It turns out, as we'll get into some details, this enzyme also has the ability to metabolize or process pesticides, insecticides that are used in agriculture. And this will sort of be an interesting turn in the paper. But before we get to that, one of the things that the authors do, in addition to sort of aiming their computational pipeline to the marine mammals and the idea of independent or convergent evolution of gene losses, is for their top hit, this PON1 gene, which encodes the paraoxonase. They measure something from their data set, the phylogenetics, the so-called DN-DS ratio. You've come across this, haven't you, Vincent, in some of our...
Totally. Yeah, totally.
Yeah. So this is a metric of gene diversification to infer something about if or how natural selection has acted in the evolution or diversification of a gene.
That's how you and Harmeet and all the ilk make their living with DNDS.
That's right. For better or for worse, this has been a really useful metric for sussing out genetic conflicts that occur. That is really rapid evolution. And so the way this works, the way DNDS works, is this is just shorthand for measuring the rates of the so-called synonymous, or for the N, the part of it, non-synonymous substitutions or mutations in codons, and dividing that by the rate of the synonymous or silent changes or mutations in codons. And so this all works because of the degeneracy of the genetic code, right? So there are the idea that there are three letters, the A, T, C's, and G's, that each encode either an amino acid or a start or a stop for a protein. And there's more than one way to sort of skin the cat. So you can basically have codons 5 or 6, for example, that might encode for the amino acid leucine. Not all have 6, but there's, you can just go to a table and actually see. So if you pick a nucleic acid substitution that does not change the amino acid, so one of those 6, for example, that might code for leucine, that's considered a silent or synonymous change. You're not changing the protein sequence in any way, and so that's why it might be considered evolutionarily. It's an assumption, but it might be considered evolutionarily silent. Mm-hmm. The top of that ratio, the non-synonymous changes, these are ones where the mutation in the nucleic acid, the codon or the base, actually does code for a different amino acid, so it's called non-synonymous change. And so just by measuring that simple ratio, which you can get from simply just comparing, say, a dozen, or in this case, 100 mammalian sequences, you can infer something about the evolutionary history. So if you have a very low DNDS ratio, that's said to be consistent with purifying selection, meaning that that you have an abundance of silent changes and sort of a paucity of non-synonymous substitutions. So the idea is that any mutation that changes the amino acids will be purified out of the population because it will be deleterious. And that's kind of, even the definition of the way we think about mutations, it almost has this connotation of being a negative thing. And that's probably the majority, or one idea is the majority of substitutions out there are that way. Now, on the other end of the spectrum, an elevated DN-DS ratio, where your non-synonymous changes are in abundance over the silent ones, this can be a sign of positive selection. And that's, as you're mentioning, Vincent, that's where for a lot of immune functions encoded by genes, where we see these elevated DN-DS values. And this has been really a useful tool to infer where genetic conflicts have occurred between things like viruses in the immune system or other microbes that have competing interests from a fitness standpoint.
And you do this on a codon basis, right?
Yeah, that's exactly right. And so that's how you determine the three letters. That's how you determine whether it's silent or non-synonymous.
And you can say, this particular codon has a high ratio DN-DS, which, as you said, suggests it's been positively selected, and then you do your magic and start to change it and so forth.
Yeah, exactly right.
And then you confirm it and get a paper in PNAS. Yeah.
Not too bad for a day's work, right? So here, the authors are actually using DN-DS, but for a slightly different reason, and that is kind of the point in between. Where DN equals DS. So the ratio would be 1, meaning that the rates of the non-synonymous to the silent changes or synonymous changes are equal. And that's consistent with this sort of situation or scenario of relaxed constraint, meaning that it's just kind of floating. You're not so, and this is, you see this, you would predict this for a gene that's lost its function, that's become a pseudogene.
Right.
It's no longer encoding a function. And so if a mutation shows up, it won't be deleterious because there's no protein. Yeah. And it also won't be advantageous because there's no protein, which might have an adaptive change if there was one. And so what you'd predict from theory is that DN-DS over some amount of time after a gene is pseudogenized would be exactly 1. Now, in practice, it does turn out to be a little bit tricky to discern between an elevated DN-DS and just sort of a relaxed constraint DN-DS of 1. There's some more details to that. But in this context, it becomes sort of a nice way of validating or just having sort of an orthogonal view of whether or not this enzyme PON1 might be a pseudogene. And so what you can see from there, again, from that phylogenetic tree, this is figure 1C. If you look at the 3 lineages of marine mammals, the independent ones, you see this brightly colored increase in DN-DS hovering around 1 for the species that they indicate that have lost the PON1 gene. Now, in contrast to that, a lot of the gray branches on the tree, these are ones that have a low DN-DS. So that's consistent with purifying selection. The idea that the gene is conserved, and that if you make mutations that would change the coding sequence, that would change the amino acids in the peptide and the protein, that would be deleterious. Right. And so they see that for most of the terrestrial mammals, that the gene looks like it's under purifying selection. It's being kept around or conserved for a valuable function. Perhaps in metabolism to keep lipid levels at sort of in check so that your cholesterol doesn't go through the roof. You can imagine that would have a big fitness consequence if you had a heart attack before you were able to have offspring.
Yeah, but afterwards it doesn't matter, right?
Well, that's a topic for another day.
It is.
Active area of work as well, right? Yep. Okay, so then this gets actually back now to they've shown it's been independently lost. You can go in and see, they confirm from the genome scale data that if they go to individual species and sort of resequence that the gene appears to be lost, the DN-DS ratios are hovering around 1, consistent with it being a pseudogene. But then they point out among those pinnipeds, so that was the seals and the walruses and sea otters, that it's a little bit of more variable scenario. So every one of the whales and dolphins have lost the PON1 gene. Mm-hmm. Both manatees and dugongs have lost it. However, for the seals and walruses, the pinnipeds, some have lost it and some haven't. So it looks like the spotted seal, the walrus, the first seal, and still have it, whereas the monk seals, Weddell seals, and sea otters have lost it. So they kind of they correlate that with actually the lifestyles of these critters. So the seals that have lost it are the ones that sort of on a day-to-day basis are doing these deeper dives or spend more time in the depths of the ocean. Whereas the first seals and the walruses are more terrestrial kind of in their behaviors. And so they don't make a big point of it, but just that might be what's going on here with the loss, that perhaps, so there's two sort of main explanations that you might, or hypotheses that you might consider for why this thing is lost. PON1, the enzyme, is lost so often. One is that there could be some cost to it, especially perhaps for the deep divers, that it actually sort of, you want some level of phospholipids. The diet of those animals is very different. The kinds of fats that they're exposed to, certainly versus terrestrial, and then even versus the more terrestrial marine mammals. They might want phospholipids to not be processed as part of their metabolism somehow. Or it could be, I think the main alternate hypothesis could be what, they've sampled about a dozen different marine mammals and maybe, 10 or so of these pinnipeds, but the N is still pretty small here. It could just be that it's lost, kind of a use it or lose it scenario.
Right.
And it just is still kind of swirling around the drain, so to speak.
Yeah, this is a long time ago that these were lost. And you can imagine that if you don't particularly need a gene and it sustains a mutation like the ones we're talking about here, it doesn't matter, right? So,
yeah, that's right. Yep. Yeah. So whatever, I think that's probably some ongoing work, and you might have even sort of expected that the paper might go that direction, that they would continue to sort of probe which hypothesis. But I think what's clear is that it hasn't been a one-off event, that most marine mammals lack the PON1 gene. And then here's where the paper takes a bit of a turn. So the idea is that the ancient loss of PON1 might have some real consequences for life in the oceans today. And in particular, the fact that more and more chemicals, more and more pollution are getting into the oceans, including a very specific set of pesticides or insecticides, this class of the so-called organophosphates, including one that's been in the news recently, chlor-pyri-fus, which is a pesticide that is used, I think, for fruit trees and orchards, so apples and peaches and so forth, to try to keep insects from eating them. This is a compound which PON1 can metabolize, meaning that it's less likely to have any. And I should say the action of the pesticide is it's a neurotoxin, which has a big influence on insects that can't metabolize it, and they get basically wiped out if they are in contact with it. But there's increasing evidence that some of these organophosphates might have some consequences, not just for insects, but for other species. And because it's known that the PON1 gene codes this enzyme that can detoxify it, the fact that all marine mammals basically are lacking PON1 sort of sounds a potential alarm bell. Could it be that as we start using more and more organophosphates for agricultural purposes, as these things run off into the oceans or related waterways, could this have neurotoxic effects on marine mammals, many of which, as we all know, are endangered and in some cases highly endangered species already?
Now, you know what Dixon de Pommier would say to this. Should be farming indoors and you wouldn't have any runoff.
That's right.
Because you don't have to use any pesticides. Thank you.
Yeah.
My God.
You have a physical barrier. Yep.
You don't have to use a pesticide if you're gardening indoors because the pests don't get in there. Therefore, you don't have runoff. You don't even need that much fertilizer because it's hydroponically grown. So that would be another solution. Now, let me ask you, Nels.
Yeah.
This activity on the organophosphate compounds is completely coincidental because these are man-made compounds, right?
Yeah, that's exactly right. And so the physiologic function is to hit these phospholipids that we find in our bloodstream, and they get to elevated levels based on the foods that we're eating. But you're right. And so as any, with any technology, we're sort of jumping from one potentially toxic compound to another. So, if we were back 50 years, we might be talking about DDT and the massive impact that had, sort of side effects it had.
Right.
This is sort of another kind of a step forward. So it's a trade-off where getting rid of DDT, for example, to go after needing some insecticide coverage. And then now what we're discovering is we've probably created a new problem. And so it's this kind of constant theme of, advancing technologies where you might sort of get past an issue that's bad on one hand, but you don't quite solve it altogether, and you've sort of created a new issue, and you keep hopping from solution to solution.
But, it's just remarkable that you couldn't predict this because the pesticides are pretty recent additions. So
that's right. Yep. No, that's right. And this is where this research, I think, is really valuable and important, is that sometimes, even under the best of intentions, which is to gain some efficiencies in crops to, for example, feed the world, that you might have sort of cryptic problems that this research uncovers.
Yeah.
And so it's not just so that, it starts, as we've been discussing, with a very evolutionary or phylogenetic approach. But what the authors do, which is, I think, a really nice experiment, is actually test whether, are these gene losses of PON1 associated with any phenotype? Can we do an experiment to see whether this might actually have a consequence for detoxifying the pesticide? So what they did, I think a nice set of experiments here, is they got serum from a sampling of the both marine mammals and other terrestrial mammals, and then measured the ability of the serum to detoxify increasing amounts of these organophosphate insecticides. So chlor-pyri-fus and another one, diazonase. It's a mouthful. And so what you can see in figure 2 is basically they're measuring the ability of the serum, which in the cases of the terrestrial mammals includes PON1, compared to the dolphins, manatees, etc., where they have found that the gene is lost. And what you can see is pretty striking. So all of the marine mammals that they test cannot process or metabolize any of the pesticides, whereas sheep, goats, ferrets, for example, their serum can. And a really nice comparison here is actually, so they have a wild-type mouse that encodes the PON1 gene, and then a knockout mouse. And you can see the ability go from, pretty reasonable, at least from the quantitative analysis they show, down to 0 for the knockout mouse. And that basically lines up exactly with the marine mammals. And so that offers, some functional evidence that there could be a real consequence to being able to metabolize or deal with these pesticides if you were exposed to it in a marine environment, but basically left without a means of dealing with or processing pesticide, which could then, you would infer, could have then the neurotoxic effects that are used to knock down the insects could also have side effects on the marine mammals as well.
Yeah, it's really a amazing finding. It's the kind of thing you just could not, as I said, you could not predict, and has amazing implications, right? It's just very cool. And they also refer to a mouse, a knockout mouse for PON1, and say that they reference a paper which says that it has higher sensitivity to organophosphate exposure compared to wild-type mice. So that's a nice model where you can test it directly, right?
Yeah, I agree. Nice comparison point, sort of a yardstick for what you might predict would happen.
Very cool.
With the marine mammals. Yep. So then they draw, a pretty dramatic connection in the last part of the paper to where the manatees, for example, in Florida, are living. And they show a map of Florida. They zoom in on Brevard County, and they're highlighting here on the map where there are state and federal manatee protection zones. That's shown in red. And then they show the waterways in blue, and then how these, these landmarks are juxtaposed with agricultural land shown in green. And so you can imagine that if on the agricultural land, especially if it's a fruit orchard, where organophosphates have been in use in recent years that there could be real exposures. And in fact, to the manatees that are, as we know, a threatened species. And some sampling of those, they mention, lift up some evidence of some of the aquatic sampling in those areas, which indeed do have measurable amounts of organophosphates. And so it could be that the horses already are out of the barn, so to speak, in terms of some of the exposures. And then the question becomes, how much of an impact could this be having on these endangered species?
Mm-hmm. So, Nels, could the reverse happen? Could the presence of these organophosphates select now for marine mammals where the function of the gene is restored?
Yeah, it's a great question. And, in best case scenario, you'd imagine that somewhere lurking in that population, maybe that they haven't sampled, maybe there are some that have held on to it. Here's where, I don't want to throw too much cold water on that idea, but so could there be a reversion, or is there sort of some existing standing variation where the gene is still intact? So even in the title of the paper, right, and then sort of implied in the phylogenetic analysis is this idea of ancient convergent losses. And then when you couple that, to that elevated DNDS or the DNDS ratio near 1, what that sort of implies, and you could, look at this directly, is basically that as soon as you have a debilitating mutation, given enough time, these pseudogenes just continue to acquire debilitating mutations. And so if it's true that that loss event is ancient enough and sort of covers the whole population, and then if you have, more than one mutational steps, in some cases maybe even dozens of mutational steps, if there's frame shifts and the gene is just sort of degrading right before your eyes in the genome, then the prospects of a reversion are kind of exceedingly low, because you'd almost have to go back and make X number of mutational steps to regain it.
Right.
And when you kind of consider some of the small genome or population sizes, I should say, the bottlenecks for some of these endangered species, there's not a ton of genetic variation out there to sample on to begin with. So ideally, you would hope, if the levels were high enough and the fitness costs were bad enough, that might come in, and that would be sort of the next step in kind of the adaptive race here. But it's possible that you've hit a dead end because these genes have been lost so long ago. And then it's only in the kind of the last blink of an eye, the last few years, that we've really been using the organophosphates in sort of massive quantities that could really be sort of...
It's not going to happen in 5 years. It's going to take thousands of years, especially with these small population sizes, right?
Yeah, that's right. And so it's sort of this difference between kind of selective pressure. If you're, if you're within 10 feet of a volcano as an entire population, and if you have a few heat resistant genes, you might be sort of a cold comfort at that point.
On the other hand, if, as we said earlier, if this happened to be a random event that was just lost because it wasn't needed, use it or lose it, that would be good, because it would mean there's no negative consequence for getting it back.
That's right. And so you could imagine I think you could imagine like a real emergency scenario where you have just a handful kind of zoo specimens for some of these species. Unfortunately, we're heading that direction for some of the marine mammals, where you could imagine trying to then intervene with a genetic sort of approach, even a gene modification or a genetic engineering approach in those highly endangered species. That would be, we're talking super last ditch efforts here, and kind of, I don't know, that might be another, sort of what's the phrase, fiddling with the deck furniture on the Titanic kind of scenario. But,
right. It seems to me that the manatees are the main issue here, right? They're right by the coast in Florida where a lot of this runoff occurs. And I'm not sure that they're so easy to genetically modify and work within the lab.
No, correct. We're talking about a pretty crazy idea. There's also, the river dolphins in some of the freshwater rivers. I think those are highly endangered as well. And so, it's not mentioned in the paper, but one thing that caught my eye, and maybe you noticed this too, Vincent, if you look at the, those DNDS ratios on that tree, there's another lineage that lights up quite a bit that actually does have the PON1 genes. And this is our own species. So humans and all of the other primates.
Yeah, that's right.
And so what that implies, so a couple things so the gene is still there. So it's certainly not a pseudogene, but what that's hinting at is that there is some standing genetic variation, kind of in the last 35 million years in the sort of descent of the primates. And so again, this could be a couple of things happening. One is this idea of positive selection, that there's something going on where the variation is being selected for, that there's some advantage to it. Or it could be this, it's again hard to discern between relaxed constraint. You can just tolerate mutations or substitutions and still have the same sort of functional output.
Yeah.
Now, if we actually think about not just sort of the marine mammals, but even our own species. So I don't know if you saw this in the news, even in the last few weeks, but there is emerging scientific evidence that these organophosphates could be having a negative impact on humans that are exposed to it. So people in the orchards working, harvesting apples and peaches and so forth, and especially if it's children. So I think there's evidence of, because this is a neurotoxin, of developmental delays, some impact, for example, on IQ tests. I haven't looked closely at all of the data, but the EPA, Environmental Protection Agency, the scientists, in part being spurred by environmental groups, have recommended that some of these pesticides, including the chlor-pyri-fus, be banned. Now, you might, I don't know if this caught your eye, but Scott Pruitt, who was the EPA chief up until recently, he overruled the scientists and overruled the ban, and so that currently these organophosphates of these classes are being used actively up until even in the last week. Kind of an interesting coincidence with this paper emerging, where a federal judge actually instituted a ban on some of these pesticides. And so this is sort of an ongoing policy meets politics meets science, not just for marine mammals, but actually for our species as well. And so one of the things that makes me curious is whether or not that sort of signal of increased variation in our species could actually have functional outcomes. Are there some humans, for example, that might be more resistant or sensitive to these things? It's maybe not, but it's something that it just illustrates how little we know.
Yeah, I was wondering in the plasma experiments, they didn't have a human sample. It would have been interesting to do some there, right?
Yeah, I agree with you. Yep, to just have that as a comparison point.
Yeah. I mean, and then depending on what you got, you might want to make a bigger study and look at some diversity, as you say. See what kind of range of values, because the marine mammals are pretty much zero.
That's right. Yeah, exactly. Yep. A little bit of residual activity, it looks like, in walruses, but if you really look at that tree, the walrus actually has an intact copy. So that probably correct. Whereas the ones where it's pseudogenized are basically flatlined for the ability to metabolize those substrates.
Also, bats seem to have some kinds of bats seem to have a high DNDS.
Yeah, I agree. It's somewhat elevated.
Not as high as gibbons and.
Yeah, correct. But it's right in that same range. And so, I mean, that again hints at the fact that, so actually among things like rodents, and rodents in particular, where you see a pretty strong, pretty low DNDS values. So that looks like it's been sort of locked in, if you will, with high gene conservation. I agree. The bats are also sort of an outlier or notable for having sort of an elevated level. And it'd be interesting to see if that could have functional consequences as well. So very possible the Clark lab is following up on that now. You can stay tuned.
Yeah, I bet after finding this, there are a lot of things that they're very excited about.
Yeah, absolutely. Lots to do. And I think, as it's really this great sort of, for science, really a great time to sort of think about doing this kind of evolutionary analysis at genome scale. So just as we're really kind of gearing up and seeing genome after genome from sampling a lot of the animal and other genetic diversity around us, these are all getting thrown into databases to sort of devise these computational pipelines to go in and sort of fish out signals that could, whether it's convergent evolution, elevated DNDS, a number of other population genetic signals. The algorithms are getting more and more sophisticated to take advantage of this really flood of data and to actually sort of prioritize what are the biologically interesting signals in there from the history of our ancestors, reaching back hundreds of millions of years in some cases. lots to learn as we sort of slice and dice these data sets.
And here, besides PON1, I have some interesting chemosensory receptors and other genes involved in that to look at too.
Yeah, that's right. Exactly. So that's just sort of like the first of, many possible things there.
Yep.
Yeah. Pretty cool. So Vincent, I got kind of excited about about this, using the convergent loss of genes sort of in natural history. And, you and I are always trying to figure out catchy sort of snappy titles for our podcast. And this one reminded me, it actually brought me all the way back to my freshman undergraduate genetics class. And my undergrad mentor, a fellow named Stefan Zweifel, who taught our genetics class, and he told us on the first day, I still remember it, that if you really want to understand genetics, you have to listen to Joni Mitchell's folk song, The Big Yellow Taxi. Do you know this song?
Oh, man, I'm a big Joni Mitchell fan way back. I used to listen to her all the time. I just loved her voice.
Yeah, fantastic.
Even saw her in concert once.
Oh, very cool.
Cornell. Yeah.
Oh, wow. Very cool. Yeah. So I was trolling around YouTube last night and pulled up the lyrics to Big Yellow Taxi, which has that great line, "Don't it always seem to go that you don't know what you've got till it's gone."
Oh, yeah.
And then, so I would put the link to this. There's a YouTube video of her singing it live. I think it's worth looking at. So my genetics professor, Stefan's point was that described that line, don't it always go that you don't know what you got till it's gone, describes genetics.
Yeah, for sure.
It's the mutation, right? You mutate something, you break it, and then you figure out for the first time what that thing did in the first place.
Yep.
And so that was kind of his big picture. And if you don't remember anything else from genetics class, remember Joni Mitchell's Big Yellow Taxi. So anyway, I was thinking, I'm really hustling here, Vincent, for our show. Good. I went even deeper into Joni Mitchell's song. And there's this great line in there. "Hey, farmer, farmer, put away the DDT. I don't care about spots on my apples. Leave me the birds and the bees." So I thought updating that for this paper, we might say, "Hey, farmer, farmer, put away the chlor-pyri-fus. I don't care about spots on my apples. Leave me the manatee in walruses."
Brilliant, Nels. You got a new career as a songwriter.
Well, I'm not going to hold my breath. That's generous of you.
You're going to keep your day job?
Keep the day job. That's generous of you to say. But
it's really nice. I love it.
We'll put this link to Joni Mitchell's song. And then I'm proposing for the title of this episode, TWiEVO 34, "Don't it always seem to go that you don't know what you've got till it's PON."
Bet.
After PON.
It's a long one, but love it. So when I was in Cornell, I took a comp lit class, and for the final exam, the question was about a play we had read by Henrik Ibsen, The Wild Duck. It's about a very dysfunctional family. And I forgot what the question was, but I had this idea that what he was trying to do was explain humans by looking at aberrant humans, by looking at people with really screwed up behavior of all sorts, and that will illuminate normal human behavior. And I said, it's like that in genetics.
Yeah.
We make mutations and we study them to understand what the normal situation is. And he loved it. I got an A on the
That's great.
It just came into my head, and he loved this because he'd never heard. He said, "What a great analogy. I got to use that." So.
That's cool. I like it.
But yeah, there you go. Yeah. Genetics is what you use to understand what was there.
That's right. Break something to understand what you had.
Break
it. That's right.
That's really cool. Good job, Nels.
Thanks. All right. Well, I think that covers it for today's paper. Congrats to the Clark Lab and Wynn and the rest of the crew. That was a nice one.
We have a couple of emails here we can knock off.
Sounds good. I'll start.
Go ahead.
The first one is from Carol. She's writing in regards to TWiEVO 28. So this is the genetics of skin pigmentation. We had a great guest on here and talking about that work. So Carol writes, "Biological scientist that I am not, I found this very interesting in civil, in a world that seems to be not so civil. Thank you for your kind conversation and so creative in promoting unique and needed contributions to science and life." Well, thanks, Carol. And I have to say, Vincent, so sounds suspiciously like someone I know very well. I think this is my mother writing into TWiEVO.
You're kidding. Is that her name, Carol?
Yeah, it is Carol, all right.
Nice.
And that sounds suspiciously like something she would write. Yeah. And she must have seen our link on Facebook. And actually, it's an interesting story. So my mom and some of her friends are part of this kind of emerging, I would say that's this aging women from the Midwest who travel in packs on Facebook. And they are going in, you just on Facebook, you see all this kind of the political stuff and people yelling. And and so she's part of a group. They go in around Facebook and they try to just calm the conversation down and say kind of supportive and positive things and kind of, drain the politics out of it and try to connect on all sides. And it's kind of an interesting strategy. I like to think that, my mom and her friends are out there as Russian bots are trying to throw gas on the fire. They're these sort of calm Midwestern women who are just sort of bringing the volume down and promoting sort of cooperation and positive thinking. So anyway, thanks, Mom.
I think there are people who still, to think calmly and seriously and look at things in a good way. I mean, people can differ on how they view something, but you don't have to be angry and full of hatred and screaming, right? I mean, I see some of these rallies where people are screaming and giving people fingers. You can just have a calm conversation, right?
Yeah, that's right. Well, and I think we've come to this point where the stuff that gets the most attention is the stuff that sort of screamed out the loudest. And the trick with that is that we start to forget about that big sort of middle space where actually there are great kind of conversations and people listening and sort of moving forward together. And so, adding that to the conversation as much as we can is not a bad.
Well, that's the whole goal of these microbe TV shows, right? Just to have a conversation. And, now and then we get a little heated because we're passionate, but we don't scream and yell and call each other names. We're trying to keep it civil, just as Carol says here. So I'm glad she likes our kind conversation. We try to be kind. Yeah. Thank you.
That's how she brought me up, Vincent. So
you're very kind. And I often try and provoke you and you never get angry. And I always say, well, this person is this and that. And you never say nasty things about people. She did a really good job with you. I'm always very impressed with your mannerisms now.
Well, you can thank Carol I can thank
Well,
Carol
you're perpetuating it too. All right. Anthony writes, Herman Melville books. At first, Moby Dick was a total flop. Did we mention this in a recent podcast? I guess we did.
Yeah.
But I don't remember what it was. Anyway, when Melville debuted Moby Dick in the United Kingdom in October 1851, the book reached American shores a month later. Many British reviewers dismissed it. Quote, this is an ill-compounded mixture of romance and matter-of-fact, wrote the London Athenaeum at the time. The idea of a connected and collected story has obviously visited and abandoned its writer again and again in the course of composition. But we talk about being nasty, right? Yeah. The style of his tale is in places disfigured by mad rather than bad English, and its catastrophe is hastily, weakly, and obscurely managed. Oof. And gives a link to this. That an editor should first send a novel off to reviewers so that they can demand revisions as a condition for publication is absurd. Is it really different in the realm of science? Oh, I see. We're probably talking about publication.
Pulling it back here to the science.
If a paper requires an imprimatur, why not just post it on a lab's website with the okay of the head? Reviewers can then be just that, comments by dead posters. Well, that's what the preprint servers are, Anthony, like bioRxiv, and I discovered one recently. Gates Open Research is another one.
I heard about that. Yeah. Yep.
You can put your manuscript up there, and of course people then also send it to a journal so they can get it published in a formal journal. But many people don't get it published, and it sits there on the preprint server forever, and it gets comments, and so yes, you can post it. And I agree. I think I have a lot of issues with peer review on many levels, both with manuscripts and grants. Many of us will say that peer review helps make your work better, and I would say in some cases that's true. You can pick up things that you want to fix and so forth, but and other times it can be nasty. As a colleague of mine said, And I may have mentioned this on the last TWiEVO. Peer review, the main function of peer review is so that your competitors can prevent you from publishing.
Ouch. Yeah, that's not a good outcome.
No, but it's not going to change anytime soon, unfortunately.
No, that's right. I mean, I'm definitely, when we've talked about this, but sort of an advocate for a little bit of the all of the above, as you're saying. So put it out there on a preprint server or something similar. And then still kind of entertain the peer review process. This kind of has a freshness to it to me right now. So we had a paper that we just did. Actually, this is the one, we had a fun discussion last time I was in New York, came in for an in vivo TWiEV that I sat in on. And this is our paper sort of on poxvirus evolution. So right as we were discussing it and it was launched on the bioRxiv, we also submitted it to the journal eLife. And just this last weekend, it got accepted. So here we are, 7-8 months later. And I would say overall, a very positive experience, but a few hiccups along the way in terms of, wait a minute, are we really going to ever get this thing out? How much energy are we devoting to updating this versus sort of what's the value added? In the end, I think it actually, and, if you want to look, you can compare the two documents because they'll both be viewable, right? The bioRxiv, and then will be linked to the eLife paper. And there are some major changes that I do absolutely agree improved the work substantially, to have some comments on it that were anonymous and, in some cases pretty critical. It was still overall constructive. And in this case, I'm actually feeling positive in the sense that in review, a couple of ideas in the conversation sort of emerged that are helping guide our next experiments. And so I think that might be the best case scenario, where that conversation sort of spurs an idea or a thought or helps to move the work forward even beyond that publication.
Yep.
And so anyway, it can be a mixed bag, as Herman Melville knows all the way back to Moby Dick. But yeah, hopefully we can continue to innovate and move forward so that we can sort of maximize the positives of peer review and get rid of those trade-offs like the ones you're talking about with competitors sort of trying to throw sand in the gears.
Just take that last one.
Sounds good. So Steve writes, I finished listening to TWiEVO 33, Fly-by Virus. And then tonight I ran across, and then Steve will put the link up to an article in the Atlantic. And this is actually what we were just discussing, which is at the, actually I guess before we started the show, we were talking about the cicada bugs. This is sort of working off the idea that there are, microbes that are influencing the behaviors. So this article that kind of came off on the heels of the, just as that virus that is encoded or is sort of deployed by a fungal pathogen that infects Drosophila fruit flies. Here's a interesting case in cicada bugs. And the title of the Atlantic article by Ed Young is, This Parasite Drugs Its Hosts with the Psychedelic Chemical in Shrooms. It also makes their butts fall off. So there's a great picture of a cicada, and this is one of these pathogenic fungi or potentially mind-controlling fungi. In this case, it grows in the back of the abdomen of the cicada. Really cool paper. I think it's Matt Caissan's lab, and then covered here in the Atlantic. And so, yeah, super cool. Thank you, Steve, for highlighting that. I highly recommend, if you enjoyed the virus sort of mind control through potentially a virus that is deployed by a fungal pathogen, you'll also really enjoy the cicada story and others like that, where we're figuring out how microbes can really control the behavior of hosts as part of their replication or life cycles.
Mm-hmm. All right, Nels, what about some picks?
Let's do it. So my science pick of the week is a local entry. So another new dinosaur species was recently discovered in Utah. This one is called Racaniello johnsoni. And I've got a link here to a short video clip, about 4 minutes, describing the discovery in southern Utah. And then sort of this cool connection to sort of local volunteer labor at our local natural history museum. So you were here, Vincent, not too long ago when we recorded a TWiEVO, or a TWiV, or TWiX, I guess, video episode with some of my colleagues.
Yeah.
and we toured around the Natural History Museum. Do you remember actually walking past? There's sort of a workshop where there are people
I did.
just kind of scraping away at dinosaur bones. Yeah.
I saw in the video they're there scraping away, and it's amazing. They're taking off the stone, trying to get down to the bone itself. It must be so hard. They say it takes hours and hundreds of hours to do this.
That's right. It's just meticulous work. And so there are a number of folks who just volunteer their time to hang out in that workshop at the Natural History Museum. This is about a short walk, maybe 15, 20-minute walk from my lab. And if you go there during the day, you can check out all of course, the dinosaur skeletons that have already been articulated. But then there are people actively working at so they'll excavate some of these fossil skeletons, but keep a lot of the rock to try to preserve or save all of the bones so that they can be articulated and then put together. And so this happened. I think this story might go back 5-10 years to a discovery in southern Utah. And in that video interview is a master's student who this was his master's degree, was working on this specimen that was discovered. And then one of the curators, who's also on the faculty here at University of Utah, just kind of describes the project. And then John Sonai, that's the last name of one of those volunteers who hangs out at the Natural History Museum,
cool.
And has ended up spending hundreds of hours pulling the dirt and the rocks away from the bones of this guy. It's a really interesting dinosaur. So the head has all of these spiky sort of protuberances on it, and it's this class of dinosaurs that have these like massive club tails, not known to be in these parts in Utah. So I think that's part of the novelty of the discovery, and obviously sort of loosely ties back to our theme of evolution. Now, in this case, what was happening on the Earth around us here 75 or so million years ago.
It's a great museum. If you're ever in Salt Lake, you got to check this out. It's really very nice.
Yeah, great to have that kind of local thing. And then it's fun, just even in the local paper from time to time, there'll be a story about a new dinosaur that they pulled out and brought up. And I love to just kind of walk over at lunch and check it out. So, yeah. How about you, Vincent? What's your science pick of the week?
So I'm reading at the moment The Song of the Dodo by David Quammen. And this,
nice.
So David Quammen is a science writer. He's also written Spillover, a few other books he's from Montana. Had the good fortune to do TWiEVO with him a couple of years ago in Boston.
Mm-hmm.
And this book is wonderful. It looks at the development of the theory of natural selection in terms of the people who did the story. That is, of course, Charles Darwin and Alfred Russel Wallace, who doesn't get a lot of press, but really kind of scooped Darwin.
That's right.
Published this stuff first. And so as Quammen once said to me, if you want to tell a story about science, tell the story of the people who did it. And that's exactly what he does here. He mostly focuses on Wallace. And, the whole thing is by studying the biogeography of islands, this is how we came to understand natural selection, because these islands get a little collection of animals and then they break away from the mainland or something happens, and you can really focus on how they've developed differently from elsewhere.
Mm-hmm.
And the key was in the 1800s, people would have to sail to these places and spend years collecting, cataloging, sending things back to the UK. They would sell it, right? But to make money to support themselves. But they ended up looking at, this couldn't have all just been put here. That was the original creationist theory, that it was all put there. And people said, well, it's not possible. From what we're seeing, the insects and the plants and the birds and the mammals, it just couldn't have been put there. It had to have developed and that's where natural selection arose. And he tells this story as if he had interviewed these two individuals, mostly Wallace, but
cool.
A bit Darwin. And it's just great because of it. I mean, you just end up whipping through it and you love it. And that's the way he is. He's a great. Have you ever read any Quammen, Nels?
Yeah, so a little bit. So I have Spillover. I haven't had a chance to pick up Song of the Dodo though. That's and you know what, Vincent? I don't know if it came across your radar yet, but Quammen just published a new book, even in the last month or so. It's called The Tangled Tree: A Radical New History of Life. And just as you're saying, so this idea of telling the story through the people who did it. So he really focuses on this guy Carl Woos, who was at Illinois, and he was a pioneer in using molecular features or characters to put together phylogenetic trees, using ribosomal sequences. And so this idea of the 3 branches, massive branches on the tree of life, the eukaryotes, the bacteria, and the archaea, that was a lot of that work came out of Carl Woese's lab. And so I haven't read the book yet. It's just fresh out. But Quammen explores this idea that's emerging from a lot of contemporary work now, that sort of the simple view of vertical descent in a tree, it's not that simple. And in fact, it's a much more, as the title says, a tangled tree with horizontal gene transfers and all kinds of other complicating features.
Yeah, totally.
So that might be time for us to pick this up and maybe even see if we can lure Quammen for appearance on TWiEVO.
I'm sure we could, since
that'd be fun.
He knows us. And I just bought the tangled. What is it that I just tangled tree?
Tangled tree. Yep.
I just bought it. Just bought it on Kindle.
Cool.
Love it. Support David Quammen.
Yeah, me too. He's a great writer.
Yeah, it would be great for us to go up there and interview him on his Montana home, right?
Yeah, maybe we could get some fly fishing in. Maybe get Dixon and.
Yeah, that's right.
Yeah.
All right, that's TWiEVO 34, Apple Podcast, microbe.tv/TWiEVO. If you listen on a phone or a tablet, you have an app that you use to listen to your podcast, please just subscribe. It would really help us a lot to have our numbers up there. Subscribe, you get every episode free. We release them about once a month. And if you really like what we do, consider supporting us, microbe.tv/contribute. Someone noted on TWiV the other day, she said she went to support us, and she said, you only have 160 people supporting you, and you have over 20,000 followers on Facebook. She said, so I helped out, so you others too as well. $1 a month would be great.
Yeah, great.
Really
That would be great to help
Nels and I get up to Quammen in Montana.
That's right. Send us up to Montana for an interview.
And, of course, questions and comments, TWiEVO at microbe.tv. You can find Nels at cellvolution.org. He's on Twitter as L. Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Great to kick around another set of evolutionary biology with you.
Always. And I love your Joni Mitchell transmogrification.
Reference.
This video, she's there in the room, looks like there are 50 people in it. When could you do that anymore these days?
Yeah, I know it. Yeah, check out that link on YouTube.
It's amazing. Really, it's amazing. Yep. I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. They're at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.